We're first going to talk about the operation of gated channels in the membrane. And remember, these gates are just proteins that are embedded in the phospholipid bilayer. So again, those membranes have built-in potential energy like a battery, and there's an electrochemical gradient due to the differences in chemical charges. So the two main types of channels that we need to know are first a chemically gated ion channel, and it's called a chemical or a ligand channel, and it opens or closes when a chemical binds. Chapter 9, you learned about acetylcholine binding, but it could be lots of other examples. It could be a neurotransmitter like dopamine, could be a neurotransmitter like serotonin, could be a neurotransmitter like norepinephrine, or it could be a hormone, even. Regardless, something binds to the outside, like acetylcholine, which is a neurotransmitter, and um, that neurotransmitter actually is going to bind over here, not on the one to the left. This one is closed, but this one opens because that neurotransmitter binds. And it could be all kinds of different examples. Whereas the voltage gated channel is a little different. So these um, chemically gated channels are only in dendrites or cell bodies, which are somas. So that's very important to remember. The voltage gated channel is only in response to ions. So it's due to ions that open them, voltage gated. So when they open in response to ions like sodium. It could also be potassium or other ions like calcium, and they are located only in axons. So it's very important to remember that the chemical gated channels are only in dendrites or cell bodies or somas, and voltage gated ion channels are only in axons. So the first potential we're going to talk about is a graded potential. And graded potentials are only happening in the dendrite and the cell body. So these are both graded potentials that we're going to be looking at. And again, they're only in dendrites or somas, cell bodies. So at rest, remember the resting membrane potential is negative 70 millivolts. But one of two things can happen. We start to move away from the resting membrane potential. So all you need to do is look at the prefix on the word. We either have depolarization or hyperpolarization. Depolarization means that it starts to move um, it starts to change a little bit. It moves from the state of polarization and it actually starts to become less negative. So, for example, it goes from negative 70 to negative 65. It's not really a big amount, but it is due to some sodium that enters the cell. And when that happens, it increases the probability of a nerve impulse, which is an action potential. Remember, we're abbreviating it as AP. The opposite of depolarization is hyperpolarization. And in this case, it moves away from the state of polarization. So it becomes even more negative. So that's really your key here. Depolarization becomes less negative. And remember, it's only referring to the intracellular fluid, whereas hyperpolarization becomes more negative. So it could go from negative 70 to negative 75, and it reduces the probability of a nerve impulse, which is an action potential. So one of the reasons for this could be potassium efflux. The potassium is leaving the cell to go into the extracellular fluid. So we have depolarization looking like this, and hyperpolarization going the opposite direction. And both of these situations are going to depend. I'm going to zoom in so you can see this a little more clearly, and then I'll zoom back out. So it depends on the neurotransmitter or the ligand, which is the chemical gated channel at either the dendrite or the soma, the cell body. So once it binds, then one of these two events happens. If it depolarizes, we call that an EPSP, which stands for excitatory post synaptic potential. However, if it is hyperpolarizing, we call that an IPSP, which stands for inhibitory post synaptic potential. So we're going to see how these graded potentials work on this slide. Graded potentials, they decay. And what this means is they decrease only with distance. So once they spread down the membrane, they start to decrease. This is exactly the opposite of an action potential, which does not decrease at all. And so remember, these are what are called sub-threshold. Threshold is negative 55 millivolts, and they never reach that. So again, it happens on the dendrite or the soma. If we go back to the previous slide, the threshold is actually negative 55 here, and it never reaches that. So that's why we say all graded potentials are sub-thresholds. For graded potential, it's determined by the magnitude of the stimulus. So again, it can be depolarizing or hyperpolarizing. If it's depolarizing, it's an excitatory post-synaptic potential. If it's hyperpolarizing, it's an inhibitory post-synaptic potential. And it's short-lived. It has local changes. So that means it only spreads a short distance. On the images to your left, you're seeing that the stimulus occurs here, but it doesn't spread very far. Here it starts to spread a little bit and then kind of dies out, and the same with over here. The distance is only a few millimeters. If we have enough graded potentials or it's significantly strong enough, it could have an additive effect where it produces an action potential. So it's named by where the function occurs, either at the receptor or at the postsynaptic potential. And what all that means, and we'll see this coming up, is when we look at an axon terminal, that axon terminal is what is releasing that neurotransmitter. Think about what you learned in chapter 9. So it's released here. So this is the postsynaptic neuron, and this represented the presynaptic neuron. So a synapse is simply where two cells, in this case, two neurons, are actually going to meet.